And I would now like to introduce and pass it on to Dr. Brian Rood, who is our first speaker in today's session. Brian is the Kurt Newman Professor of Pediatric Neuro-Oncology and the Director of Molecular Biology and Data Science at the Brain Tumor Institute at Children's National. Welcome.
Thank you, Brigitte, and thank you to the Scientific Committee for this opportunity to speak to you today. I will, advance the slides and let's get started. So, I'm going to speak to you today about a neurologic disease focused immunotherapeutic effort to target tumor-specific antigens or fragments of proteins called peptides that are created by tumor cells and not by any normal cells. And so I have no disclosures. This work really kind of arose out of another work, as so often happens in science, where you just follow the data where it leads you. And in that other work, we were basically quantitating, trying to find out what are the most abundant proteins in different subgroups of medulloblastoma, a common pediatric brain tumor, with the idea being that those proteins would be important for its biology. And the unexpected finding was that a lot of the peptides that we are identifying actually were not canonical peptides. They didn't exist in the databases of known annotated proteins. That raised the question, well, what are these things? And so with further work, we were able to figure out that these were peptides that were kind of unintended peptides. These are the result of damaged DNA that's poorly repaired, as an example, or fusions where breakage in the DNA gets put together, but the two different pieces weren't supposed to go together, creating a fusion protein. And so we started focusing on these tumor-specific peptides with the idea being that they could serve as antigens. They could be recognized by the immune system to help the immune system differentiate tumor cells from normal cells and thereby understand what to attack. And so the advantage to these tumor-specific peptides is that because they're not recognized as self by the immune system, so it reduces the risk of autoimmunity when you start ramping up the immune system to attack these things. So that should result in less on-target, off-tumor toxicity, which should give you more of a safety margin. And none of us really believe that a T-cell therapy alone is truly going to cure cancers, certainly not solid tumors. And so it's going to be necessary to use combinatorial therapies to really juice up the immune system, ramp up its potency. But in so doing, one could increase toxicity if one is not being really specific about the target that's been selected. The other nice thing about these antigens is they were not present at the time that the immune system was being trained in infancy through the thymus gland, and such there's no central immune tolerance. Normally when the immune system is trained, if it recognizes an antigen that is a self-antigen, that T cell is removed from the repertoire and thereby is no longer present, so that the highest avidity affinity TCR receptors, or T cell receptors, are culled from the immune system. And that would not be the case with a tumor-specific peptide. One of the really key findings for potency of T-cell therapies is the ability to get other T-cells interested in a tumor. So as you start to kill a tumor, you want other T-cells in the body to sort of sniff that out and start getting involved. And that's something called epitope spreading, where the targets sort of expand over time. And so if we can target multiple ones of these tumor-specific peptides, then we would have sort of standby T-cells there ready to pick up the baton as the initial wave gets exhausted. That should also address the heterogeneity of antigen expression. We know that there's subclones within tumors. Each one's present different genetic signatures, different protein expressions, and consequently, if we can sort of cover a large range of these tumor-specific peptides, we should be able to address heterogeneity a little bit better than single antigen targets. Memory immune responses. In theory, right, this tumor arose in a person with a fully functional immune system, and as such, that tumor may kind of immunize that person against the antigens that the tumor possesses. And therefore, if we can leverage that, there may be a memory component of the immune system that we could then bring to bear. And lastly, this is personalized. So each of these antigens is really unique to a given individual person's tumor. And consequently, it is not HLA restricted as some T-cell therapies, or some immunotherapies, I should say, are. And it's not sort of limited by what is commercializable and off the shelf, if you will. So this is the group that initially worked on this project. And I first need to tell you a little bit something about a technology called mass spectrometry. So mass spectrometry is a technology that identifies all kinds of different molecules in any sort of a biofluid or tissue or anything like that. In the case of proteins, the way it works is you have a database of all known proteins and a theoretical spectra, which is sort of a signature, a fingerprint of each protein fragment, is generated in this database. The mass spectrometer takes each fragment of peptide or protein called a peptide, and it basically generates the same spectra experimentally, and then a computer program matches the two and kind of gives you a likelihood list. Okay, these are the peptide sequences that are most likely to give you a spectrum matching the one that the mass spectrometer found. And so the database matching is really critical in order to sequence peptides. The problem with that is that these tumor-specific peptides don't exist in these databases because these are unintended proteins. These are not annotated normal proteins, and so they're not in the database. And so how do you really recognize them using this mass spectrometry? And so in order to tackle that, we designed this pipeline. And what I'm showing you here is actually a recent revamping and updating of the pipeline performed by the Bioinformatics Core at Children's National, and then you see the folks that did the work right there. And so basically the way this works is we start with tumor tissue, or you can start with cell lines, which is what we did when we were sort of validating this technology. We sequence the germline DNA, the tumor RNA, and we search all of those for, okay, what are all the aberrant events? What are all the breakages? What are all the fusions? What are all the non-canonical splice junctions? What are the mutations, right? What are the things that are different about the DNA from this particular tumor compared to the reference genome, the normals? And then we say, okay, well, what if each of those things was turned into a protein? What would that protein look like? And we create a database of what all of those peptides would look like if they existed. And that database runs to the, over 100,000 of these potential proteins. And then we do the mass spectrometry, and we match the two. And we sort of say, okay, well, which of these aberrant, weird things that the DNA is predicting or the RNA is predicting, what are we actually finding at the protein level, and match those up. And then we say, okay, well, we also recognize that these could simply be yet undiscovered but normal proteins. When we started all this, we were kind of naive enough to think that every protein's been discovered. That is truly not the case. These databases are not complete. And so in order to rule out any normal yet unannotated, we did a number of things. One is to run a bunch of normal brain and cerebellum. Any peptide that we find in normal tissues that's also in these tumors, we chuck that out because clearly that's just a normal peptide. GTEx is a database of sequenced normal tissues, including fetal tissues. And so we look for those to say, hey, was this maybe a protein that was created or was normal for a developing embryo and then shut off in the adult brain, but it's still a normal protein. And so we search for all of those. And then also, for every peptide that remains in this list, we go back and say, well, what was the genetic event that gave rise to it, and is that present in any normal tissue? And that's where the GTEx also comes in. So then we basically take our database, add it to the known UniProt, like sort of human database, do the searching. If it's in the normal database, we chuck it, of course, and we get down to a list of tumor-specific peptides at the end of the day. And so this is just a section of some of the tumors that we've done. We've done a couple hundred, 160 or so. And what you're seeing here in the color-coded is what's the source of the tumor-specific peptides. And so the blue is novel junctions, right? So this is where the RNA is spliced in order to be able to make multiple proteins from a single gene. The RNA is like differentially spliced in different tissues or at different developmental stages. And so these splice isoforms, sometimes in a tumor, the junctions that actually splice are not the usual junctions. They're not the normal junctions. And so you can get bits of a gene translated into protein that weren't supposed to be. And so that's the blue. So that's the predominant source of these things, not mutations. And that's sort of the key thing. So in adult tumors, mutations kind of rule the day. That's why these tumors really exist by and large, right, is mutations is the primary driver. Pediatric tumors are very different from that. And this is just another line of evidence. There's many lines of evidence to support that. But another line of evidence that really it's not mutations that are driving these tumors, and it's not driving the majority of these tumor-specific peptides either. The mutations are in green, kind of at the very bottom. You see very few of them. And then fusions are, as I described before, kind of breakages that get reformed in an aberrant fashion. What you're seeing in the top graph is those are the predicteds. So if you look, we're seeing tens to 100,000 of them, but after we do all of our whittling down, we get down to about 8.5 peptides per tumor. So, it's really a stringent filtering that gets down to just a few of these things. And then this is looking at just low-grade gliomas, a subset. We've done about 100 of those, and ependymomas, and in DIPG we've done as well. And the bottom line here is that basically all the tumors have this bad DNA that creates these weird tumor-specific peptides. One of the ways that we validate, okay, is this real? Is this peptide real? Is to synthesize, have a company synthesize a version, then we run that on the mass spectrometer and make sure that the spectrum matches up to what we found from the tumor peptide, just as a validation step. And then the other thing we did was to create, through machine learning, basically taking 250,000 peptides and their spectra from over 200 tumors and create like a neural network such that you could plug in the peptide sequence and it should return to you what the retention time as it flows through something called HPLC, high performance liquid chromatography. It's basically a way to dribble thump something through a column and then it basically separates it all out based on how much it likes or doesn't like water. And then so basically this was a way to validate these peptides as well. And so you're seeing sort of a nice correlation there between the predicted and the experimentally validated retention time based on the peptide sequence. So just another way to kind of validate that these peptides are what we think they are. The point of this slide, although it's kind of very small to see on the vertical axis there, are different tumors. And across the horizontal axis are the peptides, tumor-specific peptides, and they're color-coded for the source of those peptides. And what you're seeing here is that if you track across from any given tumor, almost all the peptides that you find are unique to that tumor, and you're not seeing them in multiple rows. And so again, this kind of gets back to that personalized thing, that these are really the result of aberrant events that occurred probably due to poor DNA damage repair and things like that. And so these are really unique to individual tumors. So the overall project schema there is sort of listed here, and I'll kind of come back to it when we talk about the clinical trial, just in the interest of time. But the next step of this was say, okay, great, so we can identify these peptides, but does the immune system recognize them? Does the immune system attack based on them? And so in order to do that, we basically synthesized a bunch of these peptides, and from donors, we took their blood, and we took the dendritic cells. Dendritic cells have the job of telling the T cells, hey, here's stuff that you should go after. And so we loaded those peptides onto that patient's dendritic cells so they could be presented to the T cells. And then we took the patient's T cells and we basically incubate them together with some cytokines, these molecules that kind of get the T cells revved up. And then the ones that find something that they're interested in attacking will expand and double and increase their numbers. And so at the end of the 3 rounds of this expansion, you can really increase the number of T cells that are specific for these specific targets. And then we take those and we characterize them. And so one of the ways you characterize them is then you put them in the presence of these peptides and say, hey, how activated do you get when you see this peptide? If you're thinking about a bloodhound searching for a prisoner, like how much do they bark when they actually see the prisoner, right? And so in this case, what you do is you put the peptide with the dendritic cell and the T cells, and then you measure something called interferon gamma, which is released by T cells that are activated. And what we're seeing here, in contrast to our other experiments where we use tumor-associated proteins, not tumor-specific necessarily, is that we get a lot more activation, basically. This may be evidence of that memory response that we were kind of hoping for before. SEB is your positive control. That basically just activates all T cells. Actin is a normal protein, so that's kind of just a background level. And so what we're finding is that all those little asterisks are showing that the statistically significant difference between actin and what we found in terms of the activation of T cells in response to those peptides. This is probably a little bit complicated, and I might just go through it very quickly, but there are a bunch of computer algorithms out there that will predict how antigenic or immune stimulating a given peptide would be. And so what we did is we took each one of these peptides, we broke it down into all of the possible pieces it could have, ran it through these prediction algorithms, chose the best scoring fragment of that peptide, and then compared that to how activating it actually was in those ELISpot assays I just showed you. And what we see is that the ones in gold are predicted to be weak binders by the algorithms, and yet in many cases they were among the best in terms of activating the immune system, whereas the ones that indicated no binding also could be the best. And some of those strong binders actually were not so great. And so it kind of comes down to, is the binding to the presentation machinery the best determinant of how antigenic something is, and likely not. So just kind of a word of caution that we don't fully understand how this works. We then look at, when we expand all those T cells, okay, so what do we have in that new, gamish of T cells that we've created in the lab? We look at the TCR sequencing to say, hey, how many individual clones are there, or different T cells that are interested in different types of peptides? And you can see that basically the top 10 range or come up with about 44% of the total T cell repertoire. So that's telling us that we've got some emergence of some real immunodominant clones there. When we do this with TAA T cells, tumor-associated antigens, so present on tumors in high amounts, but also a little bit present on normal cells, we get more like less than 1% for each of the dominant clones. And so this is kind of showing that these things may be a little bit more immunogenic than what we've worked with in the past. Okay, that's great. So they excite the immune system, but the immune system, does it kill tumor cells in response? And so to do this, we took tumor cells, and Samuele, who you see there in the corner and who's here in the room, created these tumor spheroids. And so they are dyed in red, whereas the T cells are not. And I don't know if it'll play, 'cause I don't really have a, oh, there it goes, okay. So you can see that the T cells are starting to proliferate here, and they're starting to kind of cover and diminish the red signal. The line graph in the upper right is showing you that at different ratios of T cells to tumor cells, you get increasing killing. And so the green and the purple lines are ratios of 5-to-1 and 10-to-1, and you can pretty much eradicate a tumor in about 26 hours. Bottom one will play, there we go. So these are also, again, two different ratios of T cells. And then we're left with the question, do we just cover them up with T cells so we can't see them anymore? So then we just suck them all into flow cytometry to show that indeed, right, the doubling of the labels there, indeed we did kill the tumor cells. Okay, so all of this has led to a clinical trial called IMPACT, which stands for Immunotherapy for Malignant Pediatric Brain Tumors Employing Adoptive Cellular Therapy. And so this is an active and enrolling clinical trial, and this is kind of the overall schema. So I've described most of the top of this to you, which is basically we have a patient and we resect their tumor. We do the sequencing in order to determine what are all the aberrant things that could give rise to a novel protein. Then we do the mass spectrometry on the tumor itself. We do that matching exercise to say, okay, which ones are actually present at the protein level. Then we will take the patient's own peripheral blood mononuclear cells, those are the PBMCs. Those get separated into dendritic cells to present the antigens, T cells to see those expand and attack tumor. We have a manufacturing process that gives rise to a T cell product, and then that T cell product is infused back to the patient through the intracerebral ventricular route. So we put a reservoir on the head underneath the scalp. It's just a little rubber dome in a little chamber that has a tube, and the tube goes into the lateral ventricle, a fluid-containing ventricle, which area in the brain that we can then deliver the T cells directly to the brain. And so this is being done in children less than 5 years of age with embryonal brain tumors, so malignant primitive tumors, in a radiation avoidance strategy. So they get upfront sort of chemotherapy, backbone chemotherapy, and at the tail end of that, we'll give the T cell therapy as sort of kind of a mop-up, if you will, to catch any cells that we weren't able to get with the chemotherapy alone. And that's what is sort of described here. And then RMB is for recurrent ependymoma, another malignant pediatric brain tumor. If it was treated initially with usually surgery and radiation, and if it recurs, then we can do another surgery, remove that, which has kind of become standard of care for recurrent ependymoma. Reirradiation, also a very standard approach for recurrent ependymoma. And then while that reirradiation is going on, we're making T cells in the background, and then we'll step in and give them T cell infusions after the salvage or bridging therapy. And then when we give the therapy, it is again given through the the intracerebral ventricular route. 42 days is the dose-limiting toxicity monitoring phase, and then they get the second infusion after those 6 weeks. And then they'll get an infusion every 4 weeks, up to 8 infusions. We'll be drawing CSF, cerebrospinal fluid, from that same reservoir so we can check and see what's happening with our cells. Are they persisting? Are they expanding? Are there new T cells becoming interested? Is that epitope spreading phenomena occurring? That sort of thing. So inclusion criteria, I'm not going to go through all of this, but just to sort of tell you, for the ependymoma patients, they can be any age from 1 to 30. One sort of stringent thing is that we need to be able to put that reservoir in, so they have to have ventricles large enough to sort of get the tube in there. So that's just one kind of inclusion criteria. And then exclusion criteria is overly bulky tumors. We are always concerned with immunotherapies that we could cause a lot of swelling. And if there's a large, bulky tumor in a sensitive area and it swells a lot with our therapy, then we could, cause some harm. So we're going to focus on patients who don't have a ton of disease there. And this is just one of our early patients enrolled, and this is an example of the peptide, one of the peptides that we discover, and where it's coming from. And so this is a splice isoform that you see. So in the top, this is called a sashimi plot, that kind of arc there that's got a number that's telling you that there's 2,416 reads that are demonstrating a fusion between those two parts of the RNA. If you then look in normal cerebellum, that's not there at all. But if you look in fetal tissues at 6 weeks in the hindbrain, that occurs again. So this is what would be called an oncofetal antigen, meaning that it was present and is important for fetal development, but then is turned off in the adult, or by adult, even child, anyone past that age of development. But then it is reactivated in the tumor tissue itself, and so that would be a safe target. And then, so one of the things that is really challenging about immunotherapy, and I mentioned it just a moment ago, is that we can generate a whole lot of swelling. And that's something called pseudoprogression. And one of the issues that we have as clinicians when we employ these therapies is we don't really know what to do when a patient comes in after receiving immunotherapy and their MRI looks worse. There's a lot more edema or swelling on there. It looks like the tumor's progressing. Or is it just inflammation? The MRI does not tell us the difference. So do you keep the patient on and keep treating them? Or do you say, this isn't working and stop and let's do something different? And so it's really just kind of a poor alignment between the MRI technology that we have to track these patients and the type of therapy that we're trying to administer. But what occurred to us is if you've got tumors, cells that are making these specific peptides, if you can find those peptides in the cerebrospinal fluid, that means there's still a tumor cell in there making that peptide. And one nice thing about mass spectrometry is that it comes in a couple of different forms. One is like a discovery kind of platform. We just put it all in. It's called shotgun, and you get like a ton of results back, and it's like you're swimming in data. But you can also do it in a targeted fashion. And when you do targeted mass spectrometry, you basically tell the machine, this is exactly what I'm looking for. This is the retention time, which we mentioned a bit earlier, that you should start paying attention. And this is what the spectra should look like. This is the mass over charge ratio of the peptide that we want to find, And so really it's just it sniffs down just to find what you want. And when you do targeted mass spec, it's incredibly sensitive. It's about femtomolar sensitivity for the scientists in the room. And so what we wanted to do is develop a biomarker assay where we could use these tumor-specific peptides and targeted mass spectrometry to see if we could find them in the cerebrospinal fluid that we could therefore then track the disease, track how much is present there. And so with these patients, as I mentioned, we'll have this reservoir, we'll be taking out the CSF at multiple time points going through, and at each time point we'll be doing the mass spectrometry to find this. But does it work, right? So the first experiments that we did here was with CSF, we deplete the most abundant proteins so that the mass spectrometer doesn't get too confused looking at stuff that's like super common, and just drill down on the less abundant proteins. Those are digested with an enzyme to make little manageable fragments. Then we run it on the mass spectrometer. Since we know what we're looking for, we can actually synthesize a version of the peptide that we're looking for that is isotopically heavy. What does that mean? So peptides are made up of molecules called amino acids, and those amino acids are made up of atoms, of course. And so two of those most common atoms are carbon and nitrogen. They're like everywhere, right? People have heard of carbon-14 dating. It's an isotope of carbon. Normal carbon is carbon-12. we make these, couple of these amino acids, lysine and arginine, with carbon 13 and nitrogen 15. So they're just a little bit heavier than the normal carbon and nitrogen that exist in nature. Consequently, when you make the peptides out of those amino acids, the peptide is a little bit heavier. And that's how mass spectrometry works. It measures kind of the mass of a particle. And so if we spike those in, like add them with a little pipette, and you know, okay, we're going to now look for two peptides that look identical, but they're shifted. One's just shifted a few Daltons, right, because that's the heavy part. Then you can look for these doublets, and you know how much you put in, and so you can relatively quantitate what's already there by saying, okay, well, we put in this amount, and we're finding half of that in the shifted form, and therefore, that's the amount that's there. When you do this, you can actually absolutely quantitate the amount. This we recently published like a month ago. And so this is a patient, and so their MRI scan was showing persistent but decreasing tissue in June of 2020, and when the sample was drawn, and so the mass spectrometer is showing us that there's quite a lot of that peptide there. And then a couple of months later, there was still stable enhancement at the surgical site, but we were starting to say, well, it's probably just post-operative changes on the MRI scan. The disease looks like it's really responded, and indeed, this was the end of the chemotherapy course, or nearly, for this child, and that peptide had diminished quite a bit on the mass spectrometry. And this is the MRI of that child's. Interest of time, I'll just keep going. And then the last thing is that with all of these products, we can't really predict how well they're going to work for an individual patient, right? So this is a personalized therapy. It's designed just for you, the patient, but we can't say how well it's going to work because we don't have 100 patients who had the exact same antigens in the past, who had the same immune system to draw from, right? So since it's personalized, we need some ways to kind of predict, hey, is this going to work for you? And we want to work towards a world in which we're able to do that and sort of select what are the best antigens for you, not just which ones do we find, but which ones should we really be using. And so these patient-derived tumoroids is kind of a new platform, newish platform that's gaining popularity in pediatric brain tumors, where we take pieces of tumors and we can grow them in a dish, basically. We just chop them up into really small pieces, and this is also something that Samuel is doing in the lab. And then they basically grow and they round up. And then when they get to be a little bit too big, like a millimeter, we chop them back down to like half a millimeter to a tenth to a half of a millimeter in size. And you can, some tumors you can actually expand in this way. Other tumors just kind of sit there and grow. But the nice thing is that they maintain a lot of the other cells that live inside of tumors. And those other cells are cells that suppress the immune response. They're immunosuppressive and part of the tumor microenvironment. And so these make really nice little ways to test immunotherapies because there is some of that tumor microenvironment still present in there. And so what we're trying to do is figure out if we can kind of efficacy test the TSA T-cell therapies that we're going to administer for these children to see if they then predict the outcome of the child on the clinical trial, and if so, then maybe we could develop a pathway to further select therapies that are most effective for a given patient. So that is pretty close to on time. And these are all the folks in the lab who did all of this work. To thank them as well as our funding sources and you for your attention, and I'll be happy to answer any questions. Thank you. Give Brigitte the first. Oh, okay.
Hi, thank you for your talk. I was curious about your manufacturing timeline, like from the time you get your peptide sequences to giving them to the DCs and T cells, and then if you check for any like exhaustion markers on the T cells after you do all your expansions.
Yes. So the timeline is, in theory, about 9 to 12 weeks to make the T cells. That was part of when we chose, normally, so let me back up a little. Normally when you do a clinical trial, for those folks who don't do this all the time, you kind of start with patients who have a disease that's recurrent. You don't normally start with upfront patients with a brand new diagnosis when you're testing kind of a new therapy. However, our concern was that a lot of malignant tumors, when they recur, they grow pretty fast, and those patients kind of can't sit around waiting for 12 weeks for you to make a therapy for them. And so that's why we started with those young children with embryonal tumors, because we knew they were going to get a standard of care backbone that's like 24 weeks in duration, give us plenty of time to make T cells. It should take us about 9 to 12 weeks to make the T cells. The second arm with recurrent ependymoma, the reason to kind of choose that particular group was twofold. One is that when ependymomas recur, they often aren't like really fast growing initially. They can still be pretty slow. And then secondly, that there is like kind of this backbone of reirradiation. And radiation takes 6 weeks to do. So you get a surgery, recover for a couple weeks, 6 weeks of radiation, recover a few weeks, kind of gets you close to the 12 weeks that we need to make the T cells. The downside to the manufacturing is that it's not all in our hands. So part of it is getting peptides manufactured, and that's like 6 weeks. When we started doing this, it was 3 to 4 weeks. It turns out when you want clinical grade peptides and you want to start using them in a clinical trial, then there's a lot more QA/QC, and it all takes a lot longer. So we're finding that, and then the sequencing stuff too. You think, well, you can get DNA sequenced in a few days, right? Not really. When you send it out, and there's now with the big machines and the lanes that handle so many samples and whatever, that you have to wait for them to fill the lane, and it just takes longer. So the manufacturing takes longer than we would like. We're working on a way to get that shorter, and Ian Liu, who's here in the room, has been working on that, and that's by using a mini gene. So instead of synthesizing peptides, which is costly and time expending, we can basically encode all of those neoantigens into a plasmid, and then use that to do in vitro transcription, making RNA, and then electroporate the patient's dendritic cells with the RNA so that the dendritic cells then make the protein themselves and process it according to their HLA type and present it on the MHC. And all of that is much, much quicker and way cheaper. Your last part of your question, did I answer there was something else? About exhaustion markers. Yes, so we characterize the products. And so you don't need to necessarily do 3 stimulations. Sometimes you get a big expansion after 2, and you get less exhaustion if you can go after 2 expansions, and sometimes we'll use those if that's the case. Our GMP is moving much more towards 2 stimulating rounds rather than 3 for that reason. But yeah, exhaustion is definitely an issue. Brigitte has a microphone, she wins.
Thank you. Yeah, I was wondering 2 things. One, the delivery into the CSF is because it's brain tumor, so you couldn't give it via IV, you wouldn't get to, I assume, where you want this to be. And the second question is what are your endpoints? How can you actually measure, I don't know if the endpoint is the benefit or feasibility, but I was just curious with every patient getting a different therapy.
Great question. So the intracerebroventricular delivery, so our previous trial was TAA T cell called REMIND, so it's tumor-associated antigens, targeting survivin, PRAME, and what was the third one? WT1. And so those were given systemically. But there was a couple studies that came out subsequently showing that you could get better trafficking of the T cells to brain tumors if given that way. So City of Hope and Seattle both give their CAR T cells intracerebroventricularly. And so the safety had been kind of demonstrated, and so we took that leap with this trial. In terms of it's a safety feasibility trial, 12 patients, so it's not really powered for efficacy. However, for those patients who have residual disease at the time of infusion, we'll certainly be tracking objective responses as a secondary endpoint.
Good morning. Thanks so much for this wonderful talk. I wanted to ask about the tumoroid 3D structures. I know you could probably give another hour-long talk on this alone, but I'm curious, what kind of scaffolding or stroma do you use that allows you to create these structures?
So actually none. So there's 2 different things. There's spheroids and tumoroids. With the tumoroids, you simply take the tissue from the operating room and chop it up really, really small. And then those will they look raggedy at first, and then they kind of round up over time. And then you can freeze them down at that point, or you can keep growing them. We've been working with a couple of other groups at UCSF and University of Michigan and CHOP, doing things like single-cell sequencing in order to say what cell types are in there. And you can see that there are microglia and monocytes and things that are mixed in there. Over about 4 to 6 weeks, depending upon the tumor type, those cell types drop out. but the tumors will remain. Tumor cells remain growing, and we've had some models that will grow for 150 days. The nice thing about these tumors is you can actually culture tumors that we've never been able to culture before, gangliogliomas, oligodendrogliomas, lots of low-grade types tumors do culture in this fashion without not as monolayers, but as these tumoroids. You do need, in many cases, it's helpful to have like a centrifugal shaker in your incubator. ROCK inhibitors also play a role. So there's like a cocktail, but happy to share that.
So that was a really nice presentation. So when I was looking at your workflow, you're primarily focused on kind of tumor-specific variants of known proteins. Have you looked beyond that and looked for completely novel proteins being produced?
Yeah, so yes, and so those are, most of the ones that we find are, like you said, like sort of novel isoforms. But in some cases you get like intron-intron splicing, you get translated introns. Many of them are 5' or 3' UTRs that are then splicing to an exon or to an intron. And so a lot of them are just completely novel. The entire peptide is novel and is not like a open reading frame spliced to something else. So we get those two. So yeah, kind of all over the map there. Some of the more challenging ones involve like repeats, for example, because it's hard to map those. And it's questionable how immunogenic they would be because those repeats kind of exist normally. But yeah, there's a whole, this term that's been coined of the dark proteome, which personally I take exception to it because it's not that dark. We've known about it for all along. And it's very measurable, unlike dark matter, which is hard to measure. But in any event, that's sort of this idea, right, that it's not, the functions of these things are not known. Therefore, they're kind of dark in that sense in terms of our knowledge of them. But all right, thank you.
Thank you, Dr. Rood.